the microspore mother cells which are present inside the pollen sac will undergo meiosis. That is what we consider as a reduction division. So when any cell is undergoing reduction division, the number of daughter cells which are formed will be 4. And here the microspore mother cell, as it is undergoing meiosis, we can consider this as a meiocyte. Any cell which is undergoing meiosis is called as meiocyte. And as we know that in meiosis, 2 times the cell will divide and also the nucleus, and resulting into 4 haploid cells, and these are called as microspores, or we can call as tetrad of microspores. Why? Because tetrad means 4 are present and all the 4 are not separated as and when they are divided. They have been binded with the help of an specialized complex organic material called as callose. The callose is a structure which is helping in attachment of the 4 microspores, and they always remain as one structure only. And each of the cell in the microspores is haploid in condition. So 4 microspores are formed, and when the callose is gradually dissolved, so what happens, from this tetrad all the 4 microspores are separated each other, and they are now called as individual microspores, or we can also call them as a pollen grains. And of course, as I said that they are already haploid in condition because they are formed as a result of meiosis, and any cell which is formed as a process of meiosis, we call that as a meiospore. So now we can call these microspores or pollen grains as meiospores. Microspore mother cell is called as meiocyte, and microspores or pollen grains are called as meiospores. And as I said, the tetrad of microspores are present. So in which manner does these microspores are arranged inside the tetrad? Let's see that in the form of diagrams here. So one is tetrahedral. In the tetrahedral form, the cells which have been divided will be arranged in this manner. This is called as tetrahedral arrangement. Actually, we can see the 3 microspores here, but one more microspore is present on other side of this 3 cells. So we call this arrangement as a tetrahedral, and this is most common type of arrangement. And sometimes they may also be arranged in linear form. Linear form means all the 4 microspores are arranged in the straight line like this. So we call it as a linear arrangement of the microspores. Sometimes they may arrange in decussate form, or sometimes in the T shape also. The T shape, the arrangement will be like this. So like this, the cells are in the form of a T shape. So like this, they are arranged in 5 different forms. So out of this, the most common is a tetrahedral, which is where the 4 cells are arranged in this manner. And of course here, between the 2 microspores, this part, which is, we call it as a callose wall, which is helping in holding the microspores in as a single unit. So these are the shapes which have been formed based on the arrangement of the microspores here. Then let's come to the next part of this one. So as we are saying, as we have written the heading as a microsporogenesis, that is genesis means formation, the formation of the microspores. Already we had discussed that the microspore mothers will undergo meiosis and converted into tetrad of microspores, and from the tetrad 4 microspores are separated. So this entire process from microspore mother cell to the formation of microspores is called as a microsporogenesis. And generally the number of spores which are formed will be 4, but exceptions are there. But of course here the most common pattern we are going to discuss here, that is a tetrad form or tetrahedral form, and where the 4 microspores are formed from each microspore mother cell. And where does this microsporogenesis taking place? So this process is taking place when the microspore mother cells are present inside the anther only, or inside the microsporangium only, or we can say that precocious formation we call it as, because as it is taking place inside the parent structure, that is a anther or the microsporangium. So this process is called as microsporogenesis, where the diploid structures are converted into haploid structures.